A review on the present and future aspects of various prokaryotic pigments and metabolites. Demonstrating anti-cancerous properties. Abstract prokaryotic organisms have always been known to produce a wide variety of metabolites and even various pigments. These pigments and metabolites are known to be associated with a wide range of properties including anti-cancerous, anti-bacterial, anti-inflammatory to name a few. The biggest problem that the mankind is facing today is the different types of cancers that have become a threat to humanity. This review mainly focuses on various prokaryotic organisms like Cyanobacteria, Actinomycetes, Staphylococcus gallinarum, Streptomyces peucetius, Serratia mercescens that are widely known to produce several anti-cancerous compounds. These compounds have been observed to demonstrate cytotoxic activity in cancer cell lines. A detailed study will thus help us to understand and design several novel anti-cancerous drugs. Keywords: prokaryotic, metabolites, pigments, cancer, cytotoxic. A complete review. This review aims to provide some understanding related to the varied range of prokaryotes known for producing a large number of various anti-cancer compounds. According to Newman and Shapiro, microorganisms have always known to produce a number of anti-cancerous compounds which include actinomycin D, dactinomycin, anthracyclines, including daunorubicin, doxorubicin, adriamycin, epirubicin, pirarubicin, idarubicin, valrubicin, various types of glycopeptides, bleomycin, fleomycin, the metosane mitomycin C, and the anthracenones, mithramycin, streptozotocin, pentostatin, 1,2. 11-hydroxyochlasinomycin A is a modified anthracycline which is produced by cloning. The genes involved for cloning are doxorubicin resistance gene and the aclavinone 1,1-hydroxylase gene DNRF from the doxorubicin producer, Streptomyces peucetius subspecies, into the achlasinomycin A producer. This hybrid compound is found to be highly effective against leukemia and melanoma 1,2. 2-amino-1,1-hydroxyochlasinomycin Y is another hybrid compound which is reported to be highly effective against tumors. Also, some innovative anthracyclines have been prepared by the introduction of DNA from Streptomyces purpurascens into Streptomyces galileus 1. Myxobacteria are large gram-negative rods that move by gliding or creeping. More than 400 compounds have been derived from these organisms, and one of the most effective anti-tumor compound is epithelone. These are most effective against breast cancers. Epithelone are known to bind and stabilize the microtubules that are most essentially needed for cell division and DNA replication. By the prevention of the disassembly of microtubules, epithelone successfully arrest the tumor cell cycle at the GM2/M phase and induce apoptosis 1, 3, 4. Cyanobacteria are gram-negative photoautotrophic prokaryotes. They are known for carrying out oxygenic photosynthesis and are most commonly known as blue-green bacteria. They are known for producing C-phycocyanin. Cyanobacterial products have long been known to possess anti-cancer, antiviral, antibacterial, immune modulatory, and protease inhibition activities. The anti-cancer activities are most widely studied. Lyngbya produces aprotoxin D which is cytotoxic to human lung cancer cells. 5. Simplocamide A, isolated from Simplica sp, exhibits potent cytotoxicity to not only lung cancer cells but also neuroblastoma cells. 6. The anti-cancer drugs mainly work as apoptotic modulators. 7,8. Apoptosis is mainly brought out by the implication of intrinsic and extrinsic signals. These can be by reactive oxygen species 8, the UV radiation 9, and also by the flavonoid quercetin 10,11. A demonstration by Martins and co-workers showed that when HL60 cells are exposed to aqueous extracts of Senecocystis sp and Senecococcus sp strains, cell shrinkage and membrane budding are evident, which indicates that the cells are developing apoptosis. 11,12. Lyngbya sp produces a macrolide called bislyngbyasid that induces apoptosis in mature osteoclasts. 13. And marine benthic anabena sp extracts are found to induce apoptosis in acute myeloid leukemia cell line. 14. The marine cyanobacteria brings about cytotoxicity in tumor cell lines mainly by the implication of several apoptotic indicators like cell cycle arrest, mitochondrial dysfunctions, and oxidative damage, and by alterations in caspase cascade 11. Bioactive metabolites like cryptophycin 52, chylithrixin A, hectachlorin and lingbeabalin B obtained from nostoc spp 15, chylithrix 16 and lingbia 17 respectively are found to induce cell cycle arrest in G2/M phase in different human cancer cell lines. 11. Delastidins 18 also exhibit a similar effect. Cyanobacterial aurelides A and B bring mitochondria fragmentation in HeLa cells and is evident by Mitotracker red staining 19. The activity of caspase-3, the most important caspase related to apoptosis, increases after getting exposed to simplistatin 120 and glycomacrolide bislingbazid 13. Interestingly, cryptophycin 1 is found to induce apoptosis in a human ovarian carcinoma cell line 21. Simplistatin 120 and elastidin 1022 initiates the phosphorylation of BCL2 which inhibits anti-apoptotic properties in human breast cancer cells. Curacin A is also a potential anti-cancer and anti-tumor agent obtained from a marine Cyanobacterium 23. Polyether metabolites, holochondrins, and caprolactins A, B are also known to possess anti-cancer properties 24. Caprolactins A and B exhibit mild cytotoxic activities and are produced by an unidentified gram-positive bacterium. Another compound Y-indomycinone, produced by a Streptomyces exhibits cytotoxicity against the Chinese hamster ovary, CHO, cell lines 23. Actinomycetes are also known to produce a large number of compounds with anti-tumor properties 25. Streptoalatiaceous hindustinus is commonly known to produce bleomycin, various glycopeptides, widely used for Hodgkin's lymphomas, squamous cell carcinomas and testis tumors 26. Bleomycin derivatives like bleenoxane, used in combination with other compounds is used to treat lymphomas, skin carcinomas, and tumors of the head, neck and testicles 27. SF2575, a fourth-generation tetracycline is found to have cytotoxic activity against P388 leukemia cells and various other cancer cells 28.1 of the most strongest known natural anti-cancer agents are anidines. They include dinemycin A, calichimycin, curdaracidin, esperamycin, and neocardinostatine 29,30. Anidines are represented by two classes of anti-tumor compounds, esperamycins and calichimycins 30, 31, 32. Epithylone are commonly known 16-member ring polyketide macrolide lactones. These are produced by the Myxobacterium sarangium cellulosum 33-34 and are most commonly used as anti-tumor compounds. They generally contain methylthiazole group attached by an olefinic bond and are mostly effective against breast cancer and other forms of cancer. Angiogenesis is an important step for the proliferation of cancer cells. Aspergillus fumigates produces fumagillin which acts as an anti-angiogenesis compound 36, 37. Eleuthorobin, discodermalide, bryostatins, delastidins, and cephalostatins are other common anti-tumor agents obtained from marine sources 25. The staphyloxanthin pigment obtained from Staphylococcus gallinarum KX912244 exhibits DNA damage protection activity against reactive oxygen species and anti-cancer activity which can be easily evaluated by cytotoxicity assay against 4 different cancer cell lines 38. Bacterial carotenoid pigments are an interesting group of compounds studied in relation to cancer biology. Carotenoid pigments are known to possess antioxidant property that helps fight the reactive oxygen species that are destructive to cellular molecules like DNA, proteins, and lipids 39. An experiment conducted by Avila et al. To isolate staphyloxanthin molecule from Staphylococcus gallinarum KX912244, a gut microbe of Bombyx mori delicia proves the cytotoxic effect of the extracted staphyloxanthin molecule against Dalton's lymphoma ascites, DLA, Ehrlich. Ascites carcinoma, EAC, adenocarcinomic human alveolar basal epithelial cells, A549 lung carcinoma, and Muse mucous skin melanoma, B16F10, and non-cancerous human fibroblast cell line, NIH3T3, cells in vitro using MTT, 3, 4,5-dimethylthias 01-2Y1, 2,5 diphenyl bromide, assay 39,40. Further, the demonstration by Claudet's ETAL41 proves that staphyloxanthin molecule scavenges free radicals with its conjugated double bonds, which further boost the antioxidant capability of staphyloxanthin 42,43,44. In another set of experiments, Lu et al. Demonstrated that the pigment staphyloxanthin is a strong antioxidant that can impart virulence character for the staphylococci, which will help to evade the neutrophil killing 45. But still there is no confirmation till date that staphyloxanthin can directly be used as an anti-cancer agent. 38. Prokaryotic pigment, from Serratia mercescens 46, is widely studied, and its role in inducing apoptosis and cell cycle inhibition of cancer cells is no longer a mystery. 47. A study exhibits the cytotoxic and anti-proliferative effects of prokaryotic against 60 human tumor cell lines, including that of lung, renal, leukemia, brain cancer, colon and melanoma. 48. Several research on this compound has helped us to obtain several novel compounds like novel prokaryotic analog 2,20-3-methoxy-10. Amyl 50-methyl 4-100-PYRRYL, dipyrrolmethine, MAMP. This possess potent cytotoxic activity towards cancer cells. 49. The methoxy group plays the most important role. 50. In anti-cancer effects that have been observed in several human cancer cell lines in vitro. 51, 52, 53, 54, 55. The cytotoxicity is also seen in hepatocellular carcinoma xenografts. 56, and in human primary cancer cells. 57. Interestingly, it has no marked toxicity toward non-malignant cell lines. 58, 53, 54, 55. A wide variety of bacterial taxa, including gram-negative rods such as Serratia rubitia, S. mercescens, Vibrio gazigenes, Pseudomonas magnesla rubra, Vibrio psychroerythrus Altieromonas rubra, Rugomonas rubra, and gram-positive actinomycetes such as Streptomyces spectabilis, Streptomyces longisporus, and Streptoverticillium rubereticuli are known to produce secondary metabolites related to prokaryotes. 59. Genetically engineered organisms have also proved to be highly beneficial in the production of various anti-cancer and anti-tumor activities. The attenuated strains of Salmonella enterica cervovartifimurium have several such properties. 60, 61, 62. Engineered Salmonella can demonstrate IL-2 expressing properties which can be used as an oncolytic agent in the highly tumorigenic B16F1 melanoma mouse model. 62. Live bacteria has long been used to treat human cancers. 63, 64, 65. Studies show that a proper and systemic administration of the facultative anaerobic bacteria like Salmonella typhimurium to tumor-bearing mice results in a cytotoxic and finally leading to an anti-tumor effect. 66, 67. Salmonella have been engineered in a manner so that it express cytokines or various angiogenesis inhibitors. 68, 69, 70. Studies by Bashal et al. Indicate that employing Salmonella for the treatment of tumor-bearing animals is associated with decreased angiogenesis and increased tissue necrosis within the tumors. 62. Which establish the possibility that the bacteria most probably accumulate in tumors, and thus they become targets of choice for use as vectors in order to deliver agents with anti-tumor activity into the tumor tissue. 62. A recent study by Lertler et al. 69 shows that a Salmonella strain expressing LIGHT, a TNF family cytokine, can be used effectively to target tumor tissue and bring about inhibition of the growth of primary, subcutaneously implanted, colon and breast carcinomas and their dissemination into the lungs. This anti-tumor activity of light-expressing Salmonella is found to be associated with the infiltration of inflammatory cells like IL-12 and IFN-. Two critical anti-tumor cytokines into the tumor tissue. 69, 71. The most effective anti-cancer drugs include bryostatins, discodermalide, eleuthorobin, and sarcodictine obtained from bacterial sources. 72, 73. Studies done by CART et al. And Golden et al. Indicate that lactobacilli and Noctilucus scintillans can demonstrate cytotoxicity and thus can be used as chemopreventive effects which are highly effective against colon and melanoma cancer 74,75. Additionally, lactobacilli is found to possess the ability to reduce the activities of nitroreductase, S-glucuronidase and azareductase enzymes in diet of rats, which is an indication that lactobacilli can lessen the incidence of development of colon cancer 75,76, 77. Experiments and comparative studies done by Bitzer et al. And Sagar et al. Indicate that the marine-derived Halomonas species strain GWSBWH8 can inhibit the growth of HUM02, gastric adenocarcinoma, HepG2, hepatocellular carcinoma, and MCF7 cell line 78,79. Surprisingly, exopolysaccharides, EPSs, and sulfated EPSs isolated from H. stenophila obtained from similar environment like Halomonas is reported for their pro-apoptotic effects on T leukemia cells, and most interesting element again is that they affect only tumor cells and do not show cytotoxicity against the normal cells. 80. Halomonas can also produce cytotoxic hydroxyphenylpyrrole decarboxylic acids, i.e., 3-4-hydroxyphenyl-4-phenylpyrrole 2,5 decarboxylic acid, HPPD1, 3,4-4-hydroxyphenyl, pyrrole 2,5 decarboxylic acid, HPPD2, and the indole derivatives 3-hydroxyacetyl, indole, indole 3-carboxylic acid, indole 3-carboxyldehyde, and indole 3-acetic acid. 81. HPPD1 and HPPD2 are both anti-tumor compounds, which function by the inhibition of 12-O-tetraidecanoylfurbal 13-acetate, TPA, induced activation of Epstein-Barr virus early antigen. It is found that the inhibitory effect of HPPD2 is more potent when compared to HPPD1 at all tested dose ratios. 81. Marine actinomycetes are nowhere behind. Their cytotoxic activities on various cancer cell lines are proved by many experiment and comparative analysis. They include members of the genera Diatia, Rhodococcus 82, Streptomyces 83, Selinospora 84,85, 86 and Marinospora 85,86. Nocardiopsis lucentensis, strain CNR712, produces 3-methylphorethylidineproline containing peptides. Among them lucentamycins A and B is found to exhibit in vitro cytotoxicity against HCT116 human colon carcinoma. 87. Thicoraline is a dipsopeptide isolated from Micromonospora marina. It can successfully display cytotoxic activity against both LOVO and SW620 human colon cancer cell lines. 88. Studies on Streptomyces species by Maskey et al. Puts forward the fact that trioxycarsins AC obtained from Streptomyces display potent anti-tumor activity against lung cell line. 89. The capacity of Planktomycetes to induce apoptosis and decrease cell growth in human and rat cell lines is evident by a set of experiments conducted by Calistro et al. The study further describes that Planktomycetes strains exhibit active bioactivity against one or both cancer cell lines, MOLM13 AML cells and PC3 prostate cancer, but not to the normal NRK kidney epithelial cells. 90. 2 Conclusions. This review aims to produce a basic understanding about the different types of prokaryotic metabolites and pigments and their effectiveness in various types of cancer. The capability of prokaryotes to produce anti-tumor and anti-cancerous activities is not new, but the current need is to have an overall understanding so that innovative new drugs and therapeutic agents can be developed to prevent cancer. Engineered Salmonella strains, products obtained from filamentous bacteria 91, marine Halomonas 92 and Planktomycetes 90 species provide a lot of hope. The development in molecular biology and the sequencing of microbial genomes promises a greater understanding of the tumor tissue microenvironment. This has resulted in a high boost of interest in using obligate and facultative anaerobic bacterial species, including Salmonella, Bifidobacterium, and Clostridium species, to control tumor growth. 63,65, 93.